Hello, good evening. This is Yasir Arfat. So today I welcome to all of you in the wonderful session of the botany. Today we are going to start the syllabus for APPSC DL syllabus, botany syllabus. Okay, APPSC DL botany syllabus, DL botany syllabus. So I will be going to teach you about some four important four units which are going to cover in our DL syllabus. So we will be going to see what is the syllabus which is asked in these units, these four units, and what are the important questions. First thing we will be going to study about the syllabus in detail syllabus of these four units in detail syllabus of these four units. Then after followed by this, we will be going to analyze and do the analysis of PYQs from these four units. Okay. So we will be going to do analysis of PYQs, analysis of PYQs from these four units. How much third thing when we are these two things very important, these two things, what are those two things? In detail syllabus and analysis of PYQs will make some line in our preparation. It will make some line in our preparation. So you know that the subject content is very depth. So if we see the APPSC DL syllabus, mostly the syllabus is asking at more than PG level, at the level of PG, postgraduate level. So if we start reading reference books, all those things, it is like ocean. We cannot complete the syllabus within a complete given time, short-term period. Why? Because the syllabus is very ocean. So we should mark a line. This line, we should mark a line. So where we can mark a line, how we can mark a line? We can mark up to line in the syllabus in particular topic. I will explain you. Like if one topic is there, how much depth we have to prepare, where we have to stop, what is the level of preparation should we stop at which level. This thing is very important while you are preparing for any competitive exams. So these two things, first thing is studying the syllabus and analysis of PYQs will help you to make up a line for in our preparation. Okay, so let us start the syllabus. So first thing, biology of diversity of a lower, actually it is a lower plant of which is also called as cryptogams. Okay, biology and diversity of a lower plants called as cryptogams. Okay, so crypto, what is the meaning of cryptogams? Gummy, cryptogamy, gummy. Crypto means hidden. Crypto means hidden. Gummy, gummy means marriage. Marriage. Hidden marriage. What is mean by marriage? Marriage is nothing but related to sexual reproduction. All the events which are occurring in the sexual reproduction. Okay. So marriage means will come to sexual reproduction. Sexual reproduction. So whenever we are talking about sexual reproduction, the most important part in the sexual reproduction is flower. Flower. Okay. So hidden flower means cryptogam. The cryptogams are those plants in which flower is hidden. Actually, flower is absent. Such a plants are called as what? Hidden flower plants are called as what? Non-flowering plant. Cryptogams are non-flowering plants. Non-flowering plants. Now, what are these non-flowering plants? You know that. So, if you see the plant kingdom, they are classified into two types. One is phanerogams, one is cryptogams. Yes or no? Kingdom Plantae. Kingdom Plantae is classified into two things. One is cryptogam non-flowering plants, non-flowering plants. Next thing is flowering plants, non-flowering plants and flowering plants. Okay, so non-flowering plants and flowering plants. So when you see non-flowering plants, the non-flowering plants are called as what? Can anyone answer what is this non-flowering plants are called as? Non-flowering plants are called as cryptogams. Yes or no? It is called as cryptogams. Then flowering plants, what they are called as? What are flowering plants are called as? Flowering plants are called as phanerogams. Yes, it is called as phanerogams. Phanerogams. Okay. Phanero means visible. Crypto means hidden. Okay. These are the two, we can say, sub-kingdoms. Okay. So this is a kingdom. So I'm writing here. This is a kingdom. Okay. So this is a sub-kingdom. Sub-kingdom. We will study in detail. Just introduction. Now, when you see the subkingdom, again, this is classified into two types. This one is classified into, again, two types. Subkingdom is classified into two types. In animals, it is called as, what it is called as? In animals, if you see the animals, it is called as, subkingdom is further. What is the hierarchy level of further thing? It is called as phylum. But now in the plants, it is called as what? It is called as division. What it is called as? Division. Okay, I'm writing here division. Division. Okay, so it is called as division. So what are the divisions we will study in phanerogams? The divisions which we'll be going to study in the phanerogams are called as, first thing is thallophyta. Thallophyta. We will studying thallophyta. We will be studying about thallophyta, which include two things, algae and fungi. Okay, algae and fungi. Next thing is bryophyta. 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 Next, we will be studying about pteridophyta. 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 Phanerogams, we will be going to study about spermatophyta. Spermatophyta. Now, again, what is thallophyta? Thallus. We will be going to study about thallus. Thallus means the plant in which it is not differentiated into true roots, stems, leaves is called as thallus. Okay, so we will be studying about here thallus. So what is the meaning of bryo? Bryo means moss, mosses. We will be studying about mosses. Pterido means ferns, feather-like plants whose leaves will be feather-like. That is called as ferns. You know, spermato means seeded plants which are giving rise to seeds. Okay, which is giving rise to what? Seeds. This is called as spermatophyta. Next, this spermatophyta is further classified into two groups. Further classified into subdivisions, or we can say divisions also. That is angiospermae and gymnospermae. What are those? Angiospermae. Angiosperms and gymnosperms. First, I will write gymnosperms. Why? Because in the evolution, first evolved plants are gymnosperms. Okay, gymnosperms. Next thing is angiosperms. Angiosperms. Okay, gymnosperms and angiosperms. Now, when it's come to here, you can see here thallophyta. Thallophyta is further classified into two types. One is algae, phycology, mycology. Okay. So phycology, algae, fungi. We will be studying about fungi, algae and fungi. Okay. So I will be explaining about in detail up to PG level standard of this syllabus. Okay. So we will be studying about first unit. If you see here, this is we are going to study about cryptograms. We have studied about cryptograms. Okay, in which flower is not visible. Crypto means flower is not visible. Okay, let us come here. So biology of the flowering plants, that is a cryptograms. Okay, cryptograms. So in the cryptograms, first thing here, biology and diversity of lower plants, that is a cryptograms. So now you see here, first thing in this unit only, along with algae, fungi, they have added one more chapter or one more unit that is called as bacteriology, microbial technique, means related to bacteria. So that is called as bacteriology. Okay, bacteriology. Bacteriology also we will be going to study. Means related to kingdom Monera, whatever it may be. Or we can also say related to kingdom Monera. Monera. Okay. So we will be going to study, okay, about bacteriology, study of bacteria. So here in the syllabus, if you see in detail about bacteriology is not mentioned, but some other than bacteriology means conventional studies of bacteria is not mentioned, but other than like cell wall, all those things are not mentioned. However, we'll study that what they have mentioned in the syllabus, we will see. Subtopics, they have, you can see here, 4 subtopics are there. 4 subtopics are there related to bacteria, which will come under bacteria. First thing is microbial techniques. What is microbiological techniques? Okay. So in the microbiology, now what is microbiology means? You know that what is microbiology? The study of living organisms which are very, very small. Micro means small. Okay. Small living organisms. How much small they are? How much small they are? Micro. Micro means if you take the scale, they are at the level of micrometer. Their study, their scale is at the level of micrometer, hence it is called as microbiology. Okay, so we will be going to study about this microbiology. 1 minute. Okay, so next thing is in this microbiology, very, very important point whenever you are studying something about bacteria, we should study, we should extract it, we should make it pure culture. Okay, when we are making it pure culture, then only we can study easily. What is pure culture? Means isolating that bacteria. Actually, if you take handful of the soil, okay, in the handful of the soil, you know, hundreds of the bacteria are present, but you want to study about particular bacteria which is there in the soil, such as rhizobium. You know that what is rhizobium? So rhizobium is a nitrogen fixing bacteria. So first important technique is isolation of this rhizobium. First we should isolate it. Yes or no? Where it is present? It is present in the soil. We know that. It is also present in the root nodules. We know that. Okay, we'll isolate it. Once we have isolated it, what we should do? We should make it the pure culture. Pure culture means it should be free from other bacteria. So that is only called as pure culture. Okay, it should be free from other bacteria. Hence, it is called as what? Pure culture. We should make it a pure culture. Pure culture of this. Okay, pure culture means isolation from other mixed bacteria. Okay, so we will be studying about what are the techniques by which we'll get this pure culture. 3 methods are there. Maybe you know that spread pour plate method, spread and streaking plate, all those things in detail we are studying. And enrichment media, we will be going to study about media, enrichment media, and different kinds of medias are there. Enrichment cultures, we will be going to study about enrichment media also. Third thing is anaerobic culture. Means what is the meaning of anaerobic? There are some bacteria which will live only in the absence of oxygen. An means absence. Okay, not present. Okay, not present. Aerobic means oxygen. In the absence of oxygen only, they will live. No, so how we can grow this bacteria in the absence of, how we'll make the absence of oxygen in the lab? We'll study about the three things. So first, thereby we will be going to complete about the microbial technique. Second thing, we will be studying very important point, that is microbes in medicines. So microbes, where they are useful. Okay, microbes in human welfare, or what, like that we can say this second subunit topic, we can call it as a microbes in a second topic, we can call as a microbes in human welfare. Microbes in human welfare. Human welfare. In the human welfare also many welfares are there, but in our syllabus only they have given three. So what are those? So first thing is about medicine, how, where it can be used, where microorganisms can be used. Can you say something like where it can be used? Here we will study about pure culture, followed by enrichment media, enrichment media, and followed by concluding with anaerobic culture. Anaerobic culture. Okay, anaerobic culture. So here in the, this is about first thing. So second thing is about microbes in human welfare. When you see the microbes in human welfare, so first where it will be useful. You know that microbes in human welfare, mostly where microorganisms are used in the medicines. As very important point is that antibiotics. So microbes are the source of antibiotics. So what are the organisms and their source? We will study this thing. And then after, microbes are used in agriculture also. Many other things are the small thing, but very important thing is antibiotics. Next thing, we will be going microbes in agriculture. Agriculture, how microorganisms can be used as agriculture. Very important thing is about biofertilizers. Biofertilizers. Microbes are used as biofertilizers, biocontrol agents, biocontrol agents, biofertilizers, biocontrol agents, okay, biopesticides, etc. Okay, so we'll be studying this thing. Okay, so next thing is environment. Environment is general, many things are there, like maybe you know that when ocean is polluted by oil, how we can remove that oil. Maybe you have studied like that. Many things we will study in this chapter. Third thing, when you see the third thing about this topic, microbial growth, how microorganisms are growing. You know that, yes. So nutritional requirement. What are the nutritional requirements for this growth of the microorganisms? Okay, third thing is about microbial growth. Third thing we will be studying about requirements for microorganisms. What it can be required? Can you say generally? Energy. Where you will get energy from? See, microorganisms are present in the environment, thousands of them. Every microorganisms cannot be grown in our own plate. Like when we are visiting any party, if we like that food, we'll eat that. If we don't like that food, we won't eat. Like that same thing, we are preparing the food for microorganisms. So it is our duty, which our own bacteria like which food. Okay, with respect to that bacteria, we should add the nutrients. Very important. So first thing, energy. Where it will get energy from? We'll study mostly. We'll add glucose, carbon source. Then nitrogen source. We will add nitrogen source. Okay, nitrogen source. And elements. Elements are very important. Micro elements. What are micro elements? We'll add micro elements and macro elements, both micro and macro. Micro elements will add, and macro elements, okay, to the media. So these are added in the form of salts, inorganic salts, inorganic inorganic salts. Okay, so we'll add this thing. So if I take, this is the conical flask. So for preparing media, I will add one by one. First I will add water, then I will add glucose. Okay, then I will add nitrogen source, then I will add salts one by one. according to its own weight. Okay, like that vitamins I will add which are required, and vitamins are very important regulators, harm, okay, growth regulators. So like that, I will add something one by one. We'll see what is the role, macro and micro elements. What is the role also? We will see. Okay, nutritional requirement of a microorganisms and methods to measure growth. Next thing we will study about measuring growth. Measure the growth. How to measure the growth? Okay, methods. Many methods are there. Counting method, direct method. Actually, two methods will be there. Direct method will be, direct method is one. Colony counting, indirect method is also there. Okay, so we will be going to see about these two things. Okay, direct and indirect method. Next, microbial ecology. So microbial ecology is nothing but here microbes in the environment. It is same related to microbial ecology. So here we will be studying about three important things. Next, first thing is genitrification, phosphorus solubilization, nitrogen fixation. Three things we will be going to study about. Next thing we will be finally concluding with microorganisms in ecology. Okay, microorganisms in ecology. In this, we will be studying about nitrogen fixing bacteria. Okay, nitrogen fixing bacteria. Next thing we will be studying about phosphorus solubilizing bacteria. Okay, phosphorus solubilizing bacteria. Here you can see phosphorus solubilization. Okay, like that, we will be going to conclude this unit. Hope it is clear. So in detail, we are just I'm showing you overview. So first we will study how to isolate the bacteria. Okay, once we will get the bacteria, how to maintain it as a pure culture. For this, some media required, enrichment media. If it is anaerobic microorganisms, what are the maintain? How we can maintain the anaerobic cultures. Then after that, microorganisms where it can be used. First thing is medicine, biofertilizers, biocontrol agents, environment, like that. One by one, we will be going to see. Third thing is requirements of microorganisms. What are the requirements which are required for growth of microorganisms, like glucose, carbon source, which is acting as a carbon source, then nitrogen source. Okay, then micro elements, what is their role? Then macro elements, what is their role? Then vitamins, what is their role? Okay, how they will roll. If we won't add one salt, what may happen? Like that, we will be seeing this thing. And measure of growth. How direct methods are there? Indirect methods also there. Then after finally concluding with this thing. So generally we have seen two things. Only two things we have seen. First thing is economic importance, followed by economic importance. Very important thing is about techniques. Some of the techniques. Okay. So techniques here. Here you can see first and fourth one is related to techniques. Whereas sorry, second one and fourth one is related to techniques. Okay, here first one and third one is related to techniques, whereas second one and fourth one is related to economic importance. Okay, now next thing we will be going to study about. Here you have seen about in the introduction bacteria completed. Once bacteria complete, we'll start our algae and fungi. Algae is called as what? Study of algae is called as phycology. So no, phyco means algae. So study of fungi is called as what? Mycology. Myco means fungi. Okay, mushroom. Is it clear? So here next unit we'll be going to study about phycology and mycology. So in the phycology and mycology, first we will study very important point is phycology, algae, thallus. We will be going to study about thallus. Okay, thallus, whether it is unicellular, in many things are there. So here unicellular algae is also there. Okay, unicellular one is there. Which one are unicellular? Which one are colonial? Okay, colonial. And filamentous are there. In filamentous also many types. Filamentous also there. Multicellular are there. Okay, many types are there. So and their example. Okay, like that we will be going to study about the thallus organization. Okay, then after followed by this thing, we will be going to study about in detail cell structure. Okay, cell structure of a algae. So we can take two things here, one unicellular structure, one multicellular structure. Anything we can we'll take the yeast cellular structure, and one multicellular, anything eulothrix, or any cell we can Spirogyra, any cell we will be multicell, unicellular cell structure and multicellular model organisms like anything Eulothrix, Spirogyra, any in the filamentous algae, one of the cell structure we will be going to study. Next very important thing is reproduction. You know, reproduction is the process, re means again, so producing its own kind of its similar kind. So here three types are there: vegetative reproduction, and its types we will be seeing. Many types are there. What are the types of vegetative reproduction? We will see sexual and its types also there. Okay, what are the types of sexual, asexual, and methods, mechanism, everything. Here asexual and its types also there. So we will be studying this thing. Next, followed by this thing, so our algae, you know that our algae is classified into 11 classes. 11 classes. Okay, algae is classified into further level 11 classes. So for classifying anything, some criteria is there. What is the criteria? We'll see. So what is the criteria? See here, classification of the algae based on pigments. Some pigments are there. So based on the pigments, we are classifying. Okay. So what is the pigments which are present? We will see that thing. First thing we will see about pigments. Their second criteria is reserved food material. Okay, reserved food material. Based on the reserved food material, in 11 classes, each class has its own reserved food material. So what is that reserved food material we will study? And flagella position. Every class has its own flagella and its arrangement, number of flagella, different types of flagella is different, like that we will be going to study. Classification. Okay, so this is the three important thing. First important thing is about what? Classification of algae based on what? Pigments. First we'll see pigments, reserve food material, and flagella. Three things, three criteria in depth we have to study. Flagella for 11 classes, pigments, reserve food material, and flagella. Okay, with respect to that, not 11 classes. Generally, we'll be covering 11 classes, but in syllabus particularly, they have given some important classes. So what are those important classes? Now we will be going to see. First thing is Charophyta. We will be going to study about Chara. Maybe you have Charophyta is there. Chlorophyta means green algae. Xantho means yellow algae. Okay, Bacillophyta. Okay, Bacillophyta is also there. Phaeophyta is there. Rhodophyta is there. Okay, like that. 1, 2, 3, 4, 5, 6 classes we will be going to study in detail. In our syllabus, they have given 6 classes. No problem. In detail, we will study this 11 classes in detail. Okay, is it clear? 11 classes, but concentrating on 6 classes. Why? Because they have mentioned some classes, 11 classes, but from that 11 classes, only few classes, 6 classes are particularly mentioned in the syllabus. Okay, chloro means green, you know that. All those things in detail we will study. Then after economic importance, finally concluding silent features means everything. Silent features means we will be starting with the, so I'm writing here silent features means what are the things we will be studying the silent features. Silent features means what we will be studying. First thing is habitat, where it will be there, habitat, followed by habitat. Second thing is habit. Okay, habit, that is also called as thallus organization. Okay, thallus organization, followed by thallus organization. Some of the examples we will study. Very important point is examples. Okay, so we will be studying about examples, followed by examples. Okay, so followed by examples. Fourth thing we will be studying about cell structure in detail. Cell structure. Okay. So in the thallus organization also in detail we are studying cell structure. We will be going to study in the cell structure. First thing we will be studying about many things are there. So first thing is cell wall is there. Okay, in detail. Okay. So in detail cell structure we will be going to study. After cell structure, next thing is we will be studying about its reproduction. Okay, what are the reproduction? Reproduction, about reproduction we will be going to study. So next thing is nutrition, we will be going to study nutrition. Okay, nutrition, whether it is locomotion or we will be studying locomotion. Okay, whether it has flagella or not, whether it is moving, how it is moving, all those things, locomotion we will be going to study. Followed by locomotion, reserve food material, means it will covered in the here only, cell structure only regarding flagella. Okay, very very important, flagella, reserve food material, reserve food material. Next thing is pigments, third thing is pigments. Okay, so these three things will be concentrating related to these three things. Next, followed by some special characters will be there. Okay, special features will be there. So we will be highlighting that special features, followed by life cycle. Very, very important point is the life cycle. Okay, life cycle. And finally, some economic importance also we'll study, and then after we'll conclude. 6 classes in our syllabus, 6 classes are there. So that 6 classes we will be going to study in detail this thing. Okay, so followed by economic importance. What are economic importance? Algal blooms. Blooms means you know that what is that, they will be forming on the fresh water, that is called as when algae is rapidly growing, this is, and they will form blooms, and they will totally cover the lake and therefore prevent the sunlight, take the oxygen, everything in detail we will study. Then algal biofertilizers, we will study how algae can be used as biofertilizers, will be algal blooms, algal fertilizers. Next thing is algae as a food. Okay, mostly food, some marine algae are there. Yes, marine algae such as brown algae and red algae, they will be used as food. Okay, feed means it is used as a food for humans, feed means for animals. Okay, which one are used as animals? Some important products we are extracting from the algae, so that will be going to study from this. Okay. Clear? So next thing is mycology. Same algae, same thing will be going to repeat about in the mycology, that is fungi. Okay, in the fungi we will be going to study about same thing. Same thing, first we will be studying about general features of a fungi. Okay, so you know that fungi are of 2 types. Okay, sorry, fungi filament, yes, 2 types we can say, 2, 3. Yes, first thing is unicellular form is there, fungi. Okay, if you see the fungi, first one is unicellular form. Next thing is multicellular form. Unicellular form is there. Next thing is multicellular form. Multicellular form is also there. Okay, we will be studying about this unicellular. Now this multicellular form, we will be studying about hyphae. Hyphae, okay, in the mycelium, and its specialization, modification, all this thing we will be going to study. Okay, so unicellular fungi, multicellular fungi, we will be going to study. General characters, same thing. General characters means habit, habitat. Okay, same thing. So unicellular, multicellular means, so I will be writing like this. Better we'll write like this. So same thing, fungi when it's come here. So first thing is habit we will be going to study. Habitat, where it is present, distribution, habitat, distribution. Okay, so followed by habitat, distribution. Second thing is about habit, thallus, that is unicellular, multicellular. Okay, in detail we will be going to study. So same like algae. After thallus, next thing we will be going to study about cell structure. Cell structure. So in detail, cell structure we will study, unicellular one, multicellular one we will take. Then we will study about reproduction, very very important. Okay, in the cell structure everything will be coming. Next, reproduction. Next we will study about nutrition. Nutrition. Okay, so then after, followed by life cycle we will study. Life cycle we will study. Then followed by economic importance. Economic importance. Okay, and therefore we'll conclude here. Okay, general features. General features we will study. So next thing is substrate relation. See, general features, characters of a fungi. Next thing is substrate relationship in fungi. Means some fungi are aerial. Some fungi are like they are growing. See here, this is the substrate. Some fungi, mycelium is growing within the substrate. Some fungi is growing in the air, like that. Substrate, how it is related to substrate, we will be studying about. That is nothing but mycelium. We will going to study about mycelium and its location. Next, ultrastructure, unicellular and multicellular organization, cell wall composition. I said nutrition. Nutrition is very, very important. Nutrition, three types are there. So what are those three types of nutrition? First thing is, see here, saprophytic, saproboic, biotropic. Next thing is symbiotic. Three things are there. We will be studying. The next, reproduction, same, vegetative, asexual, sexual. Then some important characters are there which are not seen in algae. Such thing will be seen in fungi. That is called as heterochalism. We'll study about this. Next, heterokaryokinesis, heterokaryosis. Next thing is parasexuality. Next, molecular accepts in the classification. In the algae, we have seen classification is based on some characters, pigments, reserve food material, flagella. Like here, we will be using molecular accepts in the classification. Means based on molecular biology, we are classifying the fungi. So what is that molecular accepts? We will be going to study. So I said 11 classes are there in the algae. Now we will see in the fungi how many classes are there. Okay. So fungi classifications is also there further. So in detail, we will be going to study about classes called as Mastigomycotina. First thing is Mastigomycotina. Next is Zygomycotina. You can see here. So Mastigomycotina. Next thing is Zygomycotina, Ascomycotina, Basidiomycotina, Deuteromycotina. Okay. Now, what is Mastigomycotina, Basidiomycotina, Ascomycotina, Basidiomycotina? Asco means, see here, sac-like structure is called as asco, sac-like structure. Basidium means basidium. Okay, club, ball-like structure. Club-like structure or ball-like structure is called as Deuteromycotina. Means actually in this sexual reproduction is not discovered. Sexual stage or sexual reproduction is absent. Okay, deutero. Therefore, they kept in deuteromyc. Whenever we discover the sexual reproduction, again it is replaced in any one of the following, like Mascomycotina, Zygomycotina, Ascomycotina, Basidiomycotina. So sexual reproduction is absent here. Okay. So mastigo means it is related to locomotion related. Zygo means pair. Okay. So these two things we will be studying in phycomycetes like that. Phyco, so together, but it is separated as a mastigomycotina, zygomycotina. Then after economic importance, what are the economic importance? Same thing we will study in detail about mastigomycotina, zygomycotina, ascomycotina, basidiomycotina, and deuteromycotina. Okay. Then their role in the industry we will be going to study. Next thing important is about industry. Okay, so how they are used in the industry. Same like here, uses in industry. Then medicine. Okay, so medicine means again antibiotics. Antibiotics are produced by two things. We will study about bacterial antibiotics. Second thing we will be studying about fungal antibiotics. Source from fungi, source from bacteria. Okay. So followed by food. As a food, some fungi is also used as a food like mushroom and single cell proteins. Okay, we will be studying about food. And a very, very important point is about fungal diseases in plants and humans. What are the fungal diseases are there? Maybe you have know about dandruff, skin disease, all those things are fungal diseases. So it affects the plants also, it will affect the humans also. Okay. Not only disadvantages of fungi, but we will be studying about importance. Importance is medicine, food. One more important economic important is mycorrhizae. Mycorrhizae is the association between fungi and yes, higher plants. Okay, so we will be studying about mycorrhizae. Myco, the word myco means what? Myco means fungi. Rhizae means what? Roots of a higher plants. So relationship between fungi and roots of higher plants. Roots of a plants. Which plants means higher plants like pinus, like that. Okay. And fungi as a biocontrol agent. We have studied in the bacteria also. Fungi as a like especially trichoderma is there. Okay. So trichoderma is used as a bio. Many things are the example just I am saying. So economic importance. Economic importance are very, very important. We are studying in bacteria also. We are studying in phycology also. We are studying in mycology also. Okay. So that is very, very important. So thereby we will complete its distribution, algae and fungi. Then followed by this thing, algae, fungi, we will go to bryophytes. Same bryophytes also, morphology we will study. Again, morphology means it's thallus organization, okay, structure, reproduction, life history, same thing, distribution, same algae, bryophytes we have studied, same thing we will study. And classification, very, very important. Classification we will study in the algae also, fungi also, bryophytes also, okay. Okay, so general characters, morphology, structure, reproduction, life history, distribution means, so this is nothing but first, all this thing is nothing but general characters, we can say. General features, general characters. Okay, in detail general features we will see. Followed by general feature, next thing is about life cycle. No, sorry, classification. Followed by this thing, classification. On what basis it is classified and what is the classification, how many classes are there. So that classes are also mentioned in our syllabus. So what are the classes which we'll be going to study? Followed by all important thing is economic importance. This thing only there. Anything you see, this much only there. General features, classification, classes, and economic importance. Only 4 things. Not much. Okay. So morphology, structure, reproduction, life history, distribution, all things will come coming in the general features. Okay, means related to all bryophytes. Then classification, 3 classes: Hepaticopsida, Anthocerotopsida. Next thing is what? Bryopsida. Like the 3 classes are there. Okay. So in that classes also, orders. Orders are of many types. See the orders. First thing is Markensialis. Okay. Next thing is Jungermannialis, Anthocerotolis, Spagnalis, Funeralis, Polytrichalis. Like that, they have given order here. Alis means orders, not classes. They have given order. Here also, in the algae, if you see, algae, fungi, phyta, they have given chlorophyce is the classes. Phyce, we are not studying about the same thing. Phyce, they have given phyta. Here, mycotina. Okay. So what is the difference between mycetes, mycotina? Okay, we will see in detail. Okay, whether it is classes, order, whatever it may be, but further classes, some they have given classes, some they have given orders. We will be studying in detail in the taxonomy. Okay, so Markensialis, Jungermannialis, Anthocerotalis means they are orders. We will be studying about orders. Orders means classes only. Means orders are different category, classes are different category, but features are same. Okay. Then followed by economic importance and ecological importance. Very, very important point here it is economic importance. However, we are studying, but ecological importance are very important. So economic importance in the bryophytes are very less compared to ecological importance are more. Why? Because these are the first plants which came on the what is the terrestrial environment and played very important role. Bryophytes. These bryophytes along with lichens played very, very important role in soil formation. Soil formation means terrestrial environment. Okay. So we will be studying this thing. Next, pteridophytes. Pteridophytes also same thing. First thing we will be studying about general features such morphology, see, Now we will be very important. We came to anatomy only. Now also not only morphology in detail, cutting the stem, all those things, and we will study anatomy. Okay, then reproduction. So first thing is general features. General features, then followed by general, many things will be coming in general features. Next thing followed by classification. You can see classification. Classification we will be going to study. Okay, so followed by classification, here some classes orders also there. So like here classes are there. See here, Psilopsida, Lycopsida, Spinopsida, Pteropsida. Okay, so we will be studying here classes. Here actually it is orders. Okay, orders. Here we will be studying classification of orders or classes. Here they have given classes. Okay, classes we will be studying. Four things are there. Followed by this thing, economic importance. Can you see economic importance? No, economic importance, they are not given that much. But other than economic importance, something they have given. So what is that? Evolution of a steel. What is steel? What is steel? When you study in the anatomy, all the layers which are present, including pericycle, below the pericycle, vascular bundles. That's all. So what will come it here? Pericycle will come. Okay, after pericycle, vascular bundles such as xylem, phloem, and pith, medullary rays, all these things will be studied in a steel. Okay, so generally anatomy, if you see, it is classified into three regions. Anatomy, if you see, first thing is epidermal region, cortex region, epidermis, epidermis, followed by cortex we will be studying. In the cortex, three regions are there. Hypodermis will be there. Hypodermis will be there. Middle cortex will be there. Followed by endodermis is there. So innermost layers of the anatomy is called as what? Steel, which will mostly usefully transport of plant, water and food. Okay, this first time vascular system has evolved in a pteridophytes. So we'll be going to study evolution of a steel. Many types of steels are there. If you see angiosperm and gymnosperm, only one type of steel is that is U steel. But here many types of steels are there. We'll see. Next, heterospory. So these are very important points. So what are those? First thing is what I am highlighting here. You can see here evolution of a steel, heterospory, origin of a seed habit. These are I'm regarding general account of a fossils. Many of the plants of pteridophytes are there which are fossilized, which are available as a fossil. So we will be studying that important things. Okay. So thereby we can conclude this pteridophytes. Okay. So therefore, we have completed a first unit. What is that first unit is called as biology of a cryptogams. Yes, here I have shown you what is cryptogam. So starting with bacteria, I am including bacteria also here. Starting with bacteria, then followed by algae, fungi, bryophytes, pteridophytes, cryptogams completed. Okay, so now we will come to phanerogams, such as gymnosperms and angiosperms. Okay. I will show you important, what is that we can say questions. Okay, I will show you questions also. But in this class, we will going to see about syllabus. Okay. Syllabus is important. Questions both are important. Okay. So next thing is diversity of a, see here, seeded plants. Means gymnosperm. Angiosperms, they have not mentioned much in the syllabus, but they have mentioned about gymnosperms. Introduction and classification. Introduction means again, general features we will be studying. Same thing, classifications we will be studying. Then after that, they have given here again orders. Okay, some important orders they have given. So what are those important orders? Cycas, okay, Cycadelis, Ginkgoalis, Coniferalis, okay, Epidralis, Gitnalis, like that we will be studying how many? 1, 2, 3, 4, 5, 6 orders are there in detail. Life cycle, all those things we will be studying. Therefore, we will completing this gymnosperms only. Not that much it is not given, but very, very important point in the with respect to, we can say, their origin area, all those things. Okay. Next, we will come to taxonomy. Okay. So taxonomy, in this taxonomy, if you see, same in this chapter only it is there, taxonomy and diversity of a seeded plants. Okay, taxonomy and diversity of a seeded plants. So taxonomic hierarchy, we'll see what is hierarchy. Levels, anything. What is classification? Taxon means what? Taxa means nomi means rules, arrangement, classification. Okay, by using the rules we are doing arrangement. That is nothing but classification of a living organism, plants. That is called as what? Taxonomy. Okay, so hierarchy, we will see what are species, like obligate hierarchies are there. Okay, intermediate hierarchies are there. So what is obligated? You know that, what is that? From kingdom to species. So keep kingdom, keep pot clean, keep pot clean. We have studied in a lower studies. Otherwise, family gets sick. Family get sick. Okay, so keep means kingdom. We will be studying about kingdom. What is kingdom? Then phylum or division. Phylum is used in the case of animal. In the plants, it is used as a division. Okay, so clean means class. We'll be using class. Then order. Next thing is family. Okay, we will be studying about family, genus and species. Okay, genus and species. In detail, we will be studying. This is called as taxonomic hierarchy. Okay. Now, if I ask, what is hierarchy? Means if I ask you address, where is your address? What you will say? You will say, if you are belonging like on the earth, where on the earth? Asia, continent Asia. Where in the Asia? Indian. Where in the India? Andhra Pradesh. State is Andhra Pradesh. Where in the Andhra Pradesh? Particular district, Anantapur. Okay. Or Guntur, or particular Vizag. Where in the Vizag? Particular mandal. Where in that mandal? Particular street, village. Like that, you will hierarchy of your address is there. Here also hierarchy is there. From kingdom to species. Okay. So species, genus, family, and other categories. And what are the principles used in assigning the relationship and delimitation of the taxa and attributes of ranks. Okay, all these things we will be going to study. Followed by, there is one code. Okay, board. Like APPSC board is conducting examination for DL Botany, all the competitive level exams like that. Here one board is there, international code for botanical nomenclature, which will be dealing with the nomenclature of plants, which is giving names to the plants. So we'll be studying about these boards, their rules, what they will follow the rules in giving particular name to a plant, how they will give. Okay, like that we'll be seeing them. So followed by this thing, taxonomic tools, such as some tools are used for identification of the plants, such a plants in the classification of plants, that is called as taxonomic tools. Okay, tools means aids. We can also call aids. Okay, tools, aids, teaching aids. Board is a teaching aid. Okay, like that we'll be using some uh, models, teaching aids. Like that in the classification, some aids are there. So first thing is about herbarium. Okay, we will be studying in detail about herbarium. Herbarium followed by books. Herbarium is the place where herbarium sheets are kept. Okay, its detail we are going to study. Then floras is a book. Then histological slides, some will be there, that we will be studying. Histological slides, cytological things will be there, cell related. Histological means tissue. Histo means tissue related. Cyto means cell related. Okay. Phytochemical. Okay. Phytochemical means chemicals which are present in the plants. mostly secondary metabolites, primary metabolites. Okay. Next is serological, biochemical, molecular techniques. Okay. DNA, RNA, genes are used in the classification. Computers will be used. GIS will be used in detail. I will explain you. No need to worry. Okay. Next thing is systems of angiosperm classification. Some systems are there for classification of angiosperms. See here, angiosperms complete. Here till now, gymnosperms completed, but in the taxonomy they have given angiosperms. So what is phonetics? What is phylogenetics? Okay. Cladistic, three are important: phonetics, phylogenetics, and cladistics. Okay, these three are important. What is their importance? How we can construct a diagram, tree diagram, all these things we will be studying. Their merits and demerits also. Next, followed by these concepts of phytogeography. Phyto means plant, geography means area. Okay, so with respect to area, okay, plants, study of plants. Like endemism. What is endemism? Particular plants are present only in particular area only. They are not present anywhere. Like we'll take some areas like Western Himalayas and some plants, hotspots. Okay, hotspots we'll be studying. Next, plant exploration means research, searching the plants, discovering the plants, their economic importance, what are the metabolites which are present in the plants, what is the, like for example, one plant is there which can give a chemical called as anti-cancer drugs. Okay, we will be going to study that thing. Next thing is invasion. Some plants are invaded in our areas. We will be going to study that invasion also. Okay, plant invention plants like Parthenium is very best, water hyacinth, Icornia. Okay, and invasion and introduction we will be studying that things. Okay, is it clear? So therefore we will be concluding the taxonomy also. Okay, so next thing is about plant resource utilization. Okay, plant resource utilization means this is nothing but how where plants are used. Ethnobotany we can say. Okay, ethnobotany. Okay, this is about ethnobotany. 1 minute. Okay, ethnobotany, where plants can be used and their economic importance. We'll study first thing is origin and evolution of the plants. Okay, followed by economic importance, such as see here, botany cultivation and uses food. Okay, some plants are used as a food. Angiosperms, especially fodder crops and their examples, fibers, okay, medicinal plants, aromatic plants, oil yielding plants. So that is nothing but totally ethnobotany we will be studying. Okay, and wood they are giving, firewood, timber wood yielding plants. Okay, non-wood products such as bamboos. Okay, many things are there. So this everything is general thing, economic importance related. Okay, we will be studying. Mostly angiosperms and gymnosperms will become. Next, green revolution. You know about green revolution has made a drastic increase in the food production. That is MS Swaminathan and many other scientists, Norman E. Borlaug in the international, in the national level, that is MS Swaminathan, and they used what are the things? Irrigation, maybe chemical fertilizers, high yielding varieties. These three are important, which are responsible for green revolution. Yes or no? So what is the three things? Benefits and adverse. What are its? Okay, what are the benefits we got it? Yield is increased, but pollution also increased due to the use of chemical fertilizers. So that thing we will studying about green revolution. Okay, so next thing is some pollution controlling plants are there. So we'll be going to study about that in the next subtopic, that is plant used as a avenue trees for shades. Like big trees are there, banyan trees. Okay, shades. Next, pollution controls. Pollution control, neem, ashoka, like that. Aesthetic means which will give beauty. Mostly they are there in the roads, in between the roads. We will be studying about such a plants, what are used. Okay. So next thing is conservation. What is conservation? Protecting particular species in its own area or out of its area. That is called as conservations. Okay, so in situ conservation, if you see, so principles of a conservation, extinctions we will study. So many species has become extinct. You know that hierarchy, red data book, red data list, red data book or red data list is also there. Okay, so environmental status of a plants based on here, very important, their ICBN, international, here you have study what is international code of ICBN, international code of botanical nomenclature. Here it is International Union of Conservation of Nature and Natural Resources. Okay, so this is also one of the board, International Union of Conservation of IUCN, Conservation of Nature and Natural Resources. Okay, so we'll be going to study about these boards. Okay, and this will be giving some list which plants are in extinct in white, like hierarchy is there in red data book list also. Extinct is there, extinct in wild is there. Okay, threatened species are there, like critically endangered species, critically endangered. Okay, critically endangered species, vulnerable species, data indeficient, data deficient, like that. Some list is there. That list we will be going to study in detail. Okay. Next, followed by two methods are there for conservation, means protection, in situ conservation and ex situ. What is in situ conservation? Now, for example, one tiger is there, one lion is there. So how we can protect the lion? We can protect the lion in the forest itself. Yes or no? And we can take out lion from that forest and we can keep in some other place such as zoos. So protecting the lion or tiger in the forest is called as what? In situ conservation. Taking out the lion from there and keeping in special area, that is called as ex situ conservation. In its own place is called as in situ. In other place, that is called as ex situ. So we will be going to study about that thing. Strategies for conservation. In situ means own place in its natural environment. Okay, so that is in situ conservation. Here we will be going to study about in situ conservation. So methods, international efforts and Indian initiatives. We have taken some protected areas in India's like sanctuaries. Okay, very important. Their examples: sanctuaries, national parks, biosphere reserves, wetlands, mangroves, coral reefs. Okay, so this thing we will be studying. Ex situ conservation principles and practice such as botanical garden, field gene bank, seed bank, in vitro repositories, cryobanks. Okay, so some general account of activities of so many other than that, some boards are there we will be studying, such as what? Botanical Survey of India, National Bureau of Plant Genetic Resource, Indian Council of Agriculture, ICAR. Okay, next CSIR labs are there. Okay, and next thing is what? DBT. Okay, what is their role, how they are significant role in conservation, all those things we will be going to study, general thing. Okay, so next thing is plant development. I think this will be deal by sir. Okay, so next thing is plant development and reproduction. 1 minute. Yes, so final unit is plant development and reproductive biology. So plant development will be mostly detailed by Chandu sir only. I will let you know. If not, I will teach this thing, but I will be going to teach you about reproduction. Okay, reproduction I will be going to teach. So reproductive biology, embryology, plant embryology, we can say. So flower is the reproductive organ. In the flower, you know that what will be there. So if you see the flower, main thing, flower has 4 parts. What are those 4 parts? First thing is about male reproductive part is called as anthers, stamen, yes, stamen, pistil. Okay, stamen and pistil. Female part is called as what? Stamen Female is called as pistil, male is called as stamen. First, we'll study about male gametophyte, then about female gametophyte in detail structure. Okay, so in this you will be studying about stamen has two things, filament and anther. Okay, so general overview, if you see here, so this is the filament. Yes or no? This is filament. This one is anther. Now, if you cut the anther, it will be looking, see, I'm cutting anther like this. It microsporangium. We will be studying about microsporangium. Here in this microsporangium, its structure we will study. So here some cells are there. So such cells are called as what? Sporocytes. What it is called as? Sporocytes. Yes or no? So here one process is taking place that is called as development of a gametophyte. One is microsporogenesis, we will see. Microsporogenesis followed by microgametogenesis. Microgametogenesis. Okay, that is nothing but development of a pollen grain. Pollen grain. Once pollen grain is developed, so how it is transferred, that is called as pollination. Okay, we'll study this thing. So for male thing, female thing, what a pistil is there. So you will take pistil like this. In the pistil, ovary, yes or no? Ovules will be there. This, okay. So we'll be studying about ovule. In the ovule embryo. This is ovule we'll consider. In this ovule, okay, so we'll consider this is the ovule. Okay, in this ovule, embryo sac is also there. Embryo sac. This embryo sac development is only called as what? Megasporogenesis and megagametogenesis. Megasporogenesis and gametogenesis. Okay, we will be studying this thing. Once male gametophyte and female gametogenesis, that is embryo sac. It leads to finally embryo sac and pollen grain. Okay, so we will study about this thing. Same thing here. Anther, microsporogenesis, role of tapetum means microsporangium, pollen development, some genes, very, very important. What are the genes which are involved? We will be studying the gene expression, male sterility. Okay, male means stamen is not developed, staminoids. Okay, sperm this is also important. Sperm dimorphism, hybrid seed production. Okay, so hybrid seed production we will study. Next thing is pollen germination. What are the things which are required for pollen germination? So pollen tube growth, guidance, pollen storage. Some pollens can be stored somewhere. So we will be studying about pollen storage, pollen allergy, pollen embryos, followed by female thing. Megasporogenesis, embryo sac organization. Different types of embryo sacs are there. We will study that. And what are the cells which are present in that embryo sacs also we will study. Okay, ovule development, here types, organization of embryo sac, structure of embryo sac cells. Okay, followed by pollen pistil interactions, which are nothing but floral characteristics. Pollination mechanisms we will study. Different types of mechanisms are there. Okay, and vectors means maybe animals, maybe insects which are involved in the pollination. Okay, which will transfer the pollen grain from one place to another, maybe insects, animals, winds, water. Okay, like that mechanism. So, and self-incompatibility means it will decrease the inbreeding depression. Due to the self-pollination, inbreeding depression may happen. So some methods are there. So that inbreeding depression should not. Bisexual flowers, self-pollination should not happen. Cross-pollination should happen. So what is that? Self-incompatibility and double fertilization is the characteristic feature of angiosperms. We will be studying that. Okay. So followed by that thing, we will be, once fertilization is completed, see here, what is there? Very easy, nothing is there. First we have studied about gametes. So no, production of gametes. Gametes production. Megaspore. Once it is completed, next thing is pollination of the pollination. Then after we will be studying about fertilization. Okay, fertilization. Fertilization gives us two things. One is embryo development. Next thing is endosperm. Endosperm. This embryo and endosperm, where it is there means it is there in the seed. Then we will study about fruit. Okay, this thing only. First we will study about flower. Flower followed by gametes production in the flower, pollination, fertilization, embryo development, endosperm development, seed and fruit. Okay. So seed development and fruit growth, endosperm development during early maturation and desiccation stages, embryogenesis, embryo development and methods. Okay. Pro embryo, globular embryo, okay, heart shape embryo, matured embryo, like that stages are there. Okay. So next thing is in the endosperm, storage of protein of endosperm and embryo, polyembryony. More than one embryo is present in a seed. That concept is called as what? Polyembryony, apomixis. Okay. So these are the things. Apomixis is what is apomixis? Development of a seeds without fertilization is called as apomixis. Embryo culture, tissue culture, and fruit formation also we will study. Followed by ending the unit with this senescence and program cell death will be explained by Chandrasekhar sir only. Okay. So cell biology you will be studying in that. Followed by we will be seeing seed dormancy, overcoming of seed dormancy and bud dormancy. Okay. So this is the in detail syllabus we will be going to study about our 4 units. Taxonomy, cryptogams, taxonomy, angiosperms, okay, and gymnosperms. Plant development, sir will be teaching, then reproduction, plant resource utilization and conservation. Okay. So therefore, thank you so much. In next class, I will be coming with the PYQs. So what are the PYQs which asked in the related to this topic? I will make one video. So thank you so much. I'm concluding the class.